Mazar. I'm a co-founder and a COO at Actuate Therapeutics. I'm a biochemist by training, but I've been doing cancer biology pretty much my whole career, with a focus on discovering and developing new drugs for treating advanced cancers, mostly solid tumors. A serial entrepreneur, founded 7 companies. 3 of them are public. And Actuate is the one I'm working with now, and probably the one I'm most excited about. And we'll be talking a little bit about our drug Elraglusib today.
My name is Joe. I've been working at Lantern for 4 years. My background was in biology. Since coming to Lantern, we've been trying to do a special type of machine learning. At least the one I'm working on is very unique for machine learning. It's using molecular features of pretreatment, treatment naive, you could say, patients or samples, and then predicting whether they will benefit from a certain drug or drug combination.
Sure. So actually, it was founded, I guess, almost 10 years ago, I believe in 2015. It was a very interesting spin-out that came out of a collaboration between a very talented medicinal chemist at the University of Illinois Chicago named Elraglusib. Alan had invented a molecule, it was a GSK3 inhibitor, with very unique properties. It was different from pretty much any other GSK3 inhibitor that was out there. And one of the main features of the molecule is it had very good pharmacology, long systemic half-life, and also was able to get into cells, especially tumor cells, very, very well. Alan had been interested in developing the molecule for neurodegenerative diseases, but he collaborated with my group when I was at Northwestern to actually evaluate the drug in some cancer models. And we saw some really profound activity in those cancer models, mostly patient-derived xenografts. And based on that, we decided together to move forward with this company. So we brought in some initial founding investors, and Dan Schmidt, who became our CEO. And the company was founded, as I said, in 2015, really with a focus on developing Elraglusib in advanced cancers. So even though it's just one molecule, we have a pipeline of many different cancer indications. Right now, we're developing the IV form of the drug, but we anticipate moving to an oral version probably in the next few years. So GSK3, it stands for glycogen synthase kinase 3. I don't know that there's a 2 or a 1, but there is a 3. There's 2 isoforms, a beta and alpha isoform. We talked about Elraglusib, which used to be known as 9-ING41, as being a GSK3 beta inhibitor, but in reality it inhibits both the alpha and the beta isoform, about the same potency. Because they're so homologous in the active site that it's almost impossible to make specific, I should say isoform-specific inhibitors. That being said, in cancer, most of the things we're looking at are probably driven by the beta isoform, based on knockdown approaches and knockout approaches that validate the beta as the target. Now, as you get into sort of immunological inflammation, which may also contribute to anti-tumor activity, the alpha may start to play a role. But that being said, there's been a number of attempts at inhibiting GSK3. It's been a pretty well-credentialed target in cancer and inflammation, also in neurodegenerative diseases, but no one has ever succeeded in getting a drug approved. A number of companies in the 90s, early 2000s, developed GSK3 inhibitors that never made it into the clinic. They failed for various tox reasons, pharmacological reasons. And so the target was never really tested in patients. One exception is the Lilly GSK3 inhibitor, which did make it to phase 1. They put it into a small phase 2, and the program was terminated. We don't know the reason why it was terminated. It was a company decision. But as we looked at the pharmacokinetic data that was published, we suspected part of the reason was just poor pharmacology. And probably you weren't getting good on-target effects with that drug if you couldn't build up plasma levels sufficiently. Our inhibitor sort of checks all the boxes from a pharmaceutical development perspective. High enough plasma levels, good pharmacokinetics to ability to get into tumor cells. And so based on its profile, which is way different than all the other inhibitors, including its safety profile, in preclinical studies, we never hit an MTD. In phase 1, we never hit an MTD with the IV. And so consequently, it told us that we had a fairly large therapeutic window which to work with for the IV version of the drug. And it gave us a lot of latitude to develop it as a single agent, but more importantly, in combination with chemotherapy. So it's those observations around the characteristics of the molecule that really drove us. Because as we talked about, GSK3 is a promiscuous molecule. It interacts with a lot of things. And in cancer, you want to make sure that you are inhibiting things that are important to cancer and not touching the other activities of GSK3. Our safety data, I believe, speaks to that. Again, this has been published as a single agent. No myelosuppression, no GI toxicity. The only side effect that we see is an alteration in vision. You sort of get a change in color and perception. It is transient. It goes away after a period of hours. And in some patients, it takes a day. It's also dose responsive. And we think that's an on-target effect because you're targeting GSK3 in the eye. But that is really the only significant adverse event. Most patients, it was mild to moderate, so it didn't affect their daily life. And so we think with that sort of profile again, being different than other GSK3 inhibitors, really convinced us that this deserved to move into patients. And so far, it's looked quite good in our trials today.
I would just want to say that it's a complicated drug target. I'm looking at this picture of GSK3B as a kinase trying to draw the interactions, and it's hard to remember them all. So I think from my point of view, that's something I would like to use a computational approach with, because it's really hard to sort of sift through one by one all of the things that GSK3 can do. And Andrew, I think you used to say like it's always second to the party.
Yeah.
Like so it has like basically a lot of contextual like factors that can play a role in interpreting like what it's doing in a certain cancer cell.
Yeah, and I think that's part of its mechanism. So we know that the interactome of GSK3 probably exceeds 100 proteins right now. It doesn't mean that it interacts with 100 proteins in every cell. Those interactions are dictated by what else is going on in that cells that protein expressed. Is it activated? And very often when we're talking about activation of proteins, especially in biochemical cascades, when we're talking about is post-translational modifications, often phosphorylations. And the reason I say GSK3 is also often second to the party is in most cases, it requires a first phosphorylation on its substrate protein to already be there before it will target that protein. And so if a pathway is turned on, GSK3 can participate. It's not turned on, and that pathway is inert. And it's maybe how the biology of GSK3 is regulated, not just in pathology, but also in normal cells. You know, we're in cancer drug development, we're always trying to find a way to better identify our patients. Unfortunately, you know, we have taken the brute force approach for many years. It's changing now. But the brute force approach was hit everybody with a sledgehammer and hope that some of the people actually benefit from it. But a lot of people don't. And so what you end up doing is exposing people to a lot of toxicity that they don't need to go through because they're not going to get any benefit from the drug. And so when we were sort of introduced to Lantern, it was through a common investor in both Lantern and in Actuate. And he suggested that we speak, so we reached out to your CEO and initiated the conversation. But we were very interested in trying to use the newest molecular tools and the newest computational tools to see if we could take our data and somehow figure out why it is that patients respond and apply that very simply to trying to select the patients. I'd say that is our goal. Our goal is to try to benefit as many patients as possible while minimizing treating patients that are not going to benefit. There's always going to be between young and our goal for the drug development program. That being said, everybody in this field talks about biomarkers. You go to investors, they want to know about biomarkers. And the reality is most people don't have biomarkers. Everybody talks about it. Everybody talks about biomarker discovery. We spend enormous amounts of money on it, and the return on investment is horrible. Now you do have some drugs that are success stories, but even there it becomes nuanced. So for example, checkpoint inhibitors like, oh, we have to look for high expression of the target PD-1. That's probably true in a couple of histologies, and then later on it turned out not to matter for many. And so clearly, we need to be able to identify our patients better. So when we were given this opportunity, we jumped at it because it gave us a tool that we could not develop internally. We're a small company. Like many small companies, we have limited funding. And so a collaboration like this made sense on a lot of levels. If you guys could access our clinical data, we could access your computational approaches, molecular approaches, and hopefully the two will lead to more than the sum of the two. So.
Elraglusib is quite different in all of the associations we saw, like with similar cell lines. And we began by modeling a few cell lines which had like public RNA. We were able to sort of use pre-treatment conditions to make a prediction of what RNA would lead to better drug response at the beginning. And we did a lot of work with PDXs, like patient-derived xenografts that were cultured, and then they had an IC50 measurement, and then we'd make models to predict that. And then what was really exciting for me is we started to get clinical data. So Actuate was very unique in what I've seen for people who have done a phase 1 trial in collecting a lot of molecular information as well as other information. But for us, we were using some genomics panel, and we had about 106 patients. And the good thing was we had real clinical data. We had a good patient number. The difficult thing was these were in different cancers, and sometimes that can make things a little bit more difficult. And the real hard thing was it was using mutation data, which doesn't, like, most people will think that's like a strange thing to say, but if you look into the literature of the few people doing machine learning with molecular data, they will say mutation data just like doesn't work as well, and it turns out it's because it's sparse. So you can imagine if like the average mutation only occurs 3 times, and you have like maybe 50 responders, and then you have to divide your data up, and you maybe only have like 1 case or 0 cases of a mutation happening, then it's really impossible to get information from that. But this like led to an interesting solution to me because we ended up finding a feature engineering approach where we would group similar mutations. These could be genes that are related because they have protein-protein interactions, or they have the same functional pathway. And after doing that, like the first thing we saw was like if we just did like what I'm showing here, if you look at the number of features that are statistically significant with like a better response was like increased about tenfold once we started to do this. So that was basically just a result of getting rid of the sparsity problem and having like more information content. And like at a conceptual level, it means like you may have some like epistatic type of functions between proteins. If you have like one mutation in one patient, you can swap it out with another. It's not exactly how it works, but that's close enough. And it's almost exactly how it works. So once we did that, we were able to make good models. To be honest, before doing that, let me see if I have this up here, but like the models that we were getting before starting that, oh, I didn't put it. The models that we were making before like doing the feature engineering, they were better than random, but they weren't that great. Like, to be honest, like compared to what we did with RNA at the beginning with a smaller set of samples. A problem turned into something interesting, and relevant to this project, we were able to start looking at features and maybe try to interpret them with relationship to drug activity. We saw some things that were related to GSK3 directly, as in like GSK3 protein-protein interactions. A few other pathways that were directly related to GSK3, and we also saw some things that were of interest, such as like things related to the immune system, which, as I learned in this collaboration, there's a lot of interest within GSK3B inhibition and activation of the immune system. So the way it can be used is like, there's basically three ways it can be used. Once you have a model, you can make predictions if you have the appropriate data type. So this means like anywhere there's mutation data, which is one of the most common types of data out there, we can make a prediction using this model. We evaluated our model on a test set, and it was almost 90% accurate, which is pretty good. We evaluated on a test set, meaning that wasn't used in the training data. There's no data leakage. So sometimes that's worth clarifying, because you can get a misleading estimation of accuracy often when you hear people talk about how accurate an AI model is. But it had a really high level of accuracy, which is probably about as high as we can go. So basically anywhere there's public data or data we get from future patients, you could use this model to predict the result. And so if you found something like there's an indication out there, which I guess I'll save it for later, but if there's an indication like where you have a high rate of response and there's an unmet clinical need, it would make sense to go after that. And you can also use this to look for independent biomarkers. So when I was talking about feature engineering, one of the things that we get out of machine learning models is extraction of what the model thinks is most important. That's helpful because we prevent the model from being a black box model, which gives people a lot of uncertainty with sort of trust of machine learning. We can tell them precisely why a prediction is being made. And when we have these models, we can extract what is the most important feature, and that can act as a biomarker fairly often. We did this for a number of projects, and in this project, there's several biomarkers that work with clinical data. If you just stratify like the high or low patient feature level, then you can get a prediction of how well they're going to do. It's not as good as using the model by itself, but it's something that you could take out as an individual biomarker. And lastly, you can get sort of drug mechanisms of that. One of the features that we were talking about that came up with in sort of an unbiased way from the machine learning was related to chromatin remodeling, and Andrew was telling me this is something that makes sense from some of their studies at Northwestern in the past.
We actually started at the opposite end, and I think we converged in the middle, which is where the models met us. So our interest, of course, is being able to, number one, understand why some patients respond and some patients don't. We then like to also understand which cancer indications are more likely to respond to a drug versus others. And then ultimately, you'd like to be able to select those patients prospectively and only include the ones that have a higher probability of response in your trial. And so when we looked at this, we looked at it as an opportunity to take really a lot of data, because we had a big phase one. Our phase one ended up being, I think the total was 200 some patients, around 267 patients. That was between single agent, and the second part of the trial was combination with 8 baskets of chemotherapy. And this was published just earlier this year in Clinical Cancer Research. But we had a lot of outcomes data for those patients, number one. And for quite a few of them, we actually had molecular information, as Joseph already said. So we had sequence information, sometimes other pieces of molecular data that we could then use to create and feed the models. Now, despite the fact that we had 27 different histologies, all these patients were advanced cancer patients. Most of them had failed 3 prior treatments or more. And so it's essentially a salvage therapy. Patients that exhausted standard of care, they're not going to do very well. And so we could ask questions about a number of different clinical outcomes. The obvious one people go for is response. Does your tumor shrink or not? The reality is, when you have an advanced cancer population at phase one, it's actually rare to see tumor shrinkage. By that time, the tumor is so resistant that you don't see a lot of shrinkage. What we also collected is data of how long the patient stayed on drug, the progression, and how long they survived. And one of the things that we saw is we saw probably more than half our patients have very long-term survival. Now people will kind of poo-poo that data and say, well, a bunch of different histologies, it could have been just normal clinical course. That's partially true. But when you have patients that are all end-stage patients, and a lot of them are surviving 8 months, 9 months, a year, many patients longer, you have to think that that's more than just chance or probability. And so we thought there must be some commonality that makes these patients responsive to the drug or benefiting from the drug. And that's how we work back towards the models. And of course, a lot of that data with the outcomes then could be used for the validations, for the test sets, etc. And so then now the next goal will be, I don't want to jump ahead too far, but will be to apply this prospectively, which we can talk about in a few minutes to future trials.
As I was saying before, we often work with a treatment-naive condition, which is basically the molecular data that's taken before treatment starts. And that's obviously the rational way to go about this. A lot of people will use various types of molecular data after treatment for different analysis, but if we have it before treatment, we can actually know and make the decision at the right time in an actual clinical setting. And that goes for whether we're using the model or whether we're just using a biomarker derived from the model. So as far as our approach goes, we usually we do some standard things, but the most important thing that we usually do is feature engineering. When we work with biological data, it is basically different than every type of data set that you will be trained in, like in a machine learning course somewhere. They'll usually have what's called like a long data set that you have like a few types of features. So you don't really need to worry about selecting features too much or feature engineering in many cases. And you have maybe millions of samples or tens of thousands of samples, but in biology, that's very expensive. And you have very many features. So it's called like a wide data set. So we have to do feature engineering. Otherwise, we have a problem of overfitting, which means we get excited because we think we have like 100% accuracy, but it doesn't translate outside of whatever we're training on. So to evaluate this, we usually we'll just make a training and test set. So we do in the training set, you can have a separate validation set to sort of tweak the parameters of the model and also decide like how to do your feature engineering. And we use machine learning metrics to determine which features to include. We use something called the Shapley method, which is very common. It was developed in economics. Basically, you can think of it as a way to determine like what is the contribution of a feature in the context of an uncontrolled comparison. Like you could think of this like in an analogy to sports, like what is for like player A, like what is his contribution? And so we do this with all of the features, and then we start to sort of drop them. There's two reasons to do it. One is to avoid overfitting, and another is it's makes the model more interpretable. So if we have to like go through like 50 features that were involved in why a model came to some conclusion, it's going to be very convoluted and nobody can really explain that. So part of our approach is feature engineering, and I think that's I would say this is one of our specialties. This is something that is not done in machine learning generally, and we basically can do two things from that. One is we extract biomarkers, as I've been saying, and also we just make the models better when we give it a better pool of features to work with. So when I first started to work on this data, there was initially less accuracy on the clinical data than I was getting for past models that were using RNA. And I mentioned we did a feature engineering approach to sort of resolve that problem. But while I was going through that, I was testing a lot of different things to try to come to a solution. So I ended up making a tool that I would call a brute force tool. So it basically tries all of the different parameters. It randomly selects like which features to include. And this basically gives an unbiased way to go through the training data and come up with the features and final models. And it also has an interesting property which was just came about by chance. It wasn't part of our approach before, and that is basically when you make thousands of models, and you have a lot of different feature evaluations, you can get an average feature evaluation, which turns out to be very important because it's basically just an estimation which can have some variance if you look at a single model. So sometimes we may overestimate the importance of various features if we're just looking at a single model. And there's also a lot that are sort of missing because in the process of trying to only use a few features in a single model, you end up with a small set and you can't do much pattern analysis in that situation. So it turned out to be a very helpful thing that was useful for a general machine learning process in biological data. And yeah, with that, we make models. After we get them, we try to interpret them, see if it makes sense, and do things like predictions on public data. As I was saying, I was making this process that would randomly sort of select things and find the optimal sort of way to get to the result by brute force. But in practice, if you have something that is negatively associated, it may be an exclusion criteria, but it's not as useful as an inclusion criteria. Like if you have something that's saying like you're going to respond, you will be much better off than saying like you probably won't respond. It still has like a lot more uncertainty, even though we can get good accuracy with the model. If we want to sort of find some signature of drug response, it's much more valuable to look at positive ones. So following some suggestions by Andrew, what we were doing was first getting all of the positive features and then using those as the feature pool to do, and then we would do the feature selection and model building. And one of the interesting properties from that was, well, first it worked. It was not quite as good because there was like less features available, but it was still very effective. And it had over 80% accuracy in our test set. And it had the interesting property of not having false positives. So that means if it is predicting positive, you have a lot more certainty this is real. So you may have like less accuracy overall, but you have more confidence in your positive predictions. And that's a good thing in many cases.
There's, I think the important thing that Joseph said is a drug signature versus a tumor signature. And a lot of the negative features are often tumor signatures. So when you have mutations in RAS, or amplifications in McDermott or P53 mutations, those are negative features that are tumor signatures in general. Any tumor that has those is going to do poorly in that patient. It has nothing to do with our drug, whether they benefit from it or not. Those patients will not do well. And pretty much any drug that you test in patients with those mutations will generally not do well. Now it's changing now that people are taking drugs specifically for those mutations. But before, it was just a general poor prognostic marker. You look at it the other way, and you find mutations where now you give your drug, you take a patient that would have done poorly and flip them, so now they do well. That's a drug signature. And that's really what we were interested in trying to find out, because that allows us to then enrich for patients that will do well. And you can exclude patients that will do poorly without knowing anything about your drug. But you can't include patients that will do well unless you actually know your drug.
Yeah, so this has been an exciting collaboration. I think it was always exciting to be able to work with clinical data. Then we actually had a good team. They had a lot of collaborators that were like very intelligent and informed, and some people that have Dan Bilodeau at the Mayo Clinic and his group, and there's several groups at the Mayo Clinic at Brown and various collaborators. So it was a really interesting group that, like, personally was nice to get information from. And yeah, getting feedback on the models, like we were talking about the positive models, like that was just something I'd never thought of before. And when we were working with genomics data, this is definitely a data type that we want to work with. We went to AACR and ASCO together, and when I was at ASCO, I actually saw a lot of posters that are like, how do we use mutations data to like sort of predict results of the clinical trial? And you can see some of them are saying the same thing that I was talking about, mutation data is sparse. So there's no solution for this yet. So this kind of put us at the cutting edge of this problem. And I think by finding a solution that worked, we're able to work with a lot of other data. We could work with like, this will come up in many areas. It could work in liquid biopsy data. And you can get, instead of just like looking at one or two mutations of predefined interest, you can just like look at this in an objective way that's unbiased and high throughput. So it turned into something valuable for tool development. And when I was trying to sort of find something that worked better for this, that was when I sort of made a platform for automated machine learning, and all you have to do is put in like your outcome data and then whatever your features are, and then it will work automatically overnight. And this became basically my go-to tool that I've been using ever since. So uh, it wasn't planned that way, but it was just a side benefit. And from the perspective of radar, it's good to have like some more clinical experience with an ongoing trial instead of just like something that has been uh, maybe some data from some common public sources like TCGA. There is some good public data out there, but it's obviously very valuable to like work with something that's currently in the clinic in phase 2. Yeah, so when we were working with this mutation data, like we had a panel of about 400 genes, and this is sort of a common thing. You can find something that has like 1,500, and so we can work with that type of data if you have that, and you have uh, the more samples, the better. But if you have a reasonable amount of samples, then we can make a good model. And I think we can always find the best position for a drug. It does help if the drug is good. So yeah, I should mention that one of the good things of working with Actuate is when we were able to start to divide this into training data, and we wanted to say like this group is responder, this group is non-responders, we could roughly balance the data set. But for many drugs that we're going through phase 1, you'd probably have a very imbalanced data set that's mostly non-responders, and that will be very troublesome to work with. But for many drugs that aren't in that situation, whether you're working on a preclinical or clinical stage, we can basically work with any type of data. And sometimes this can be data that is publicly available. If you use some well-known cell lines, we can pull that from some common data sets. So as long as you have some sort of sensitivity or outcome measurement, we can make a model for you, and we can identify biomarkers and then try to see where's the best place to put the drug and what would be the best combinations and things like that.
Sure. So, our main focus now has been 2 programs. The main trial, the one that's most advanced, is the development of Elraglusib in patients with metastatic pancreatic cancer. We refer to that as the 1801 3D study. That study is now completely accrued, and we publicly disclosed an analysis of interim data back in April. We went public, as you said, and that was in our S1 and in our public information. It's also available on our website in our corporate deck. And we have promised another analysis of the interim data. And it'll be the November data, but it'll be available in December. And that will occur at approximately 70% of the patients in the control group reaching an event, which unfortunately is death, because the endpoint of the study is survival. Early next year, we anticipate having top-line data, and so the trial will read out, and we'll have outcomes data on that full patient population, which has a control arm of just the standard of care first-line treatment, which is gemcitabine and Abraxane, and then the experimental arm, which is Elraglusib plus gemcitabine and Abraxane. So we're, of course, very excited to see that data, because preliminarily it looked very exciting, and the data was in favor of the experimental arm. But remains to be seen what we see at the end of most. We've also observed that in a pediatric indication in Ewing sarcoma, we had a number of surprising complete responses. So our drug was able to eliminate tumors in refractory resistant patients. So these are kids that essentially had failed conventional treatment. Their tumors had come back, and at least 2 of those patients, we completely put them into remission. We had some other patients that had partial responses. A number of these are durable, and some of these patients are still alive. Ewing is a very rare, fortunately, it's a pediatric and it's rare type of cancer indication, but it is something we're now pursuing using the various regulatory mechanisms that help develop drugs for rare diseases and rare pediatric indications. Now going forward, that's where we really see an opportunity to apply the radar data. We are interested, both based on our phase 1 and based on the modeling, in pursuing some different indications, including metastatic melanoma, non-small cell lung cancer, possibly colorectal cancer. And in those indications, we have the ability to potentially apply some of the learnings from the radar analysis, the radar experimental approaches to that trial. How would we do that? So it's too early right now to just jump in and patient select based on that data. It has to be validated. But what we can do is we can actually put patients in like we normally would, but then stratify them for analysis based on how radar would have stratified them. And we'll see for the analysis whether using radar stratification will give us a better outcome than just using the normal type of analysis where we just include all comers. And so that'll be a way how we envision actually being able to validate that data. If it showed promise, we would then apply that to our pivotal trials. You have to talk with FDA. You have to talk about how we actually use that commercial space. But I think that'll be a good problem for all of us to have. So that's sort of the vision. I should mention that new indications are dependent on additional funding. We are not currently pursuing those. We're really focused on pancreatic and Ewing, but hopefully with future financings, we'll have the money to pursue some of these other approaches. We have a website, actuatetherapeutics.com. It's a public company, so typical public company website, but we post all our posters, all our trials, all our manuscripts and abstracts are all on the website. It also has contact information. So just any questions that anybody has or any interest in current or future trials, please reach out. We'll always respond. We have a lot of people reaching out that have interest in the program, and we're happy to talk to you about it. So,
on our website, you can see links to posters for our collaboration with Actuate, our ASCO and AACR posters, and I think soon you'll be able to look forward to a manuscript that we'll be looking to publish. And if you have interest in using radar, please reach out to us.